Blobby, slimy, crawling, changing, beautiful. The amoeba are many things, but simple and easy to understand they are not. They're some of the greatest shapeshifters in the microcosm, intensely complex, and even the same species may look very different depending on the conditions they are under. The word amoeba itself, from Greek amoebae, means change. The different appearances of amoeba are called morphotypes, and each species may have several of them. This makes the identification and classification quite difficult. And even though two amoeba might look quite similar, they can be genetically further apart than I am to an earthworm. You might recognize these guys from your time at high school in biology class, looking at pond water or a hay soup. They're super abundant in dirty water, but even more so in soil. However, it's never been possible to observe them in wild soil. We are so glad for our Soilchips, where they happily move in and out and let us study them live under the microscope.
Amoeboid organisms are all those that move in the amoeboid way. This means moving through pumping cell content into one part of the cell, pushing out their cell membranes to form pseudopodia, or false feet. These pseudopodia can have many different shapes like blobby, tapered, spiky, or branched. And their movements can look like this, or this. Some amoeba are so slow that you can only see them move through time-lapse, while some almost keep up with the ciliates. They also have the incredible ability to form cysts, creating a hard shell around their soft body to protect them from drying out or being damaged when the environment becomes dangerous for them. Cysts mostly look like little spheres, so figuring out what's hiding within them is a science all in itself. An especially eye-catching and pretty one is this one, belonging to the acanthamoeba, looking like a little star. When people think of amoeba, they often think of the most amoeba, Amoeba proteus. This little guy right here. Probably. I think he's quite spectacular. And he was first documented all the way back in 1755, so we've known about him for quite a while.
Many of the amoeba we see in our lab are in the Amoebozoa supergroup and look something like this. They are flat and ovoid, and they don't form any clear extended pseudopodia, keeping a decently consistent shape. This one is rugose, meaning it has multiple wrinkles. Sometimes we see the Limax amoeba. They don't have any wrinkles and they tend to be more slender. Limax means slug. Do you think they look like slugs?
In this next video, we have a beautiful heliozoan, which means sun animal, with beam-like pseudopodia stretching outwards from its center. They use their pseudopodia like tentacles to capture bacteria and protists that swim by, sometimes even using toxins to paralyze them before consumption. In this video, we observe a flagellate flying too close to the sun, bumping into the heliozoan, becoming poisoned, and losing control over its movement. As you can see, they can also move rather quickly when they want to. They float like a star, but sting like a bee. They're voracious predators and can clear large areas of bacteria very quickly. Some heliozoans can even fuse together into large clusters, able to capture larger prey. I saw it once and it was super cool, but I forgot to press the record button. I promise I'll show you next time.
The amoeba we've talked about so far have all been naked. But now we would like to show you a very well-dressed gentleman. He's a testate amoeba. These amoeba that build themselves shells in one way or another. They can be made of different materials like silicates, calcium, or even organic compounds. Some amoeba can even pick up pieces from their surrounding, building themselves a little house like the caddisfly larvae does. Sometimes you can see a testate amoeba that has made their house out of the beautiful shells of diatoms. Testate amoeba are incredible at building their shells. Some are so sturdy they can't be broken down by nature and are left in the soil for millions of years. These microfossils can then be studied to tell us about evolution and even about the state of the world back then.
A testate amoeba that always sticks out to us are the giant angular amoeba called monothelamids. They may even be my favorite protist. In our Soilchips we get a lot of them, and they're huge. They form a special type of pseudopodia which get extremely long, sometimes up to 10 centimeters, and they grow throughout the entire chip, fishing out bacteria. Their main body is sometimes so big that it doesn't fit into the chip at all, and then we only see its fishing rod pseudopodia reaching all the way into the chip. It's quite a scary sight, right? What's super striking about them, to me, is their ability to transport packages of nutrients and other useful things in two directions using the same pseudopodia at once. Its threads are incredibly long compared to the size of its main body. It's like if a human fisherman was throwing a 1 kilometer long fishing line to catch fish. The sickest thing is that the fishing line is also a part of the amoeba's body. Imagine how scary it would be if a spider trapped you in its web, but that web was also made out of spider. Finally, I'll show you maybe the most confusing group of amoeba, the ones that really blur the line of what is and what isn't. I'm talking about the amoeboflagellates, and they're as common as they are strange. The amoeboflagellates have a flagella but move using pseudopodia. Are they flagellates or are they amoeba? Well, they're both. You might have heard about the crazy brain-eating amoeba Naegleria fowleri. It might be the most famous amoeboflagellate. Though in our samples we find many types of amoeboflagellates, like this, but I honestly have no idea what this one.
So, we leave it here for today, at being confused and often not knowing exactly what we're looking at. I think this sentiment is strong when it comes to the soil microcosm, a wild and confusing word that science has only scratched the surface of. It's difficult and weird, but we think that's part of what makes it so flipping exciting. As always, leave a comment if you know more about what we found. Thank you for joining us for Soilwatching.
I am joined by Edith Hammer, soil enthusiast and associate professor at Lund University.
And next to me is Fredrik Klinghammer, house microscopist and the biggest protist lover I know.